Hello, everyone. Thanks for joining us today. This is the Interagency Working Group and Ocean Acidification seminar series on ocean acidification within estuarine systems. And why don't we just give just a couple more minutes for folks to straggle in. Just be advised we are going to be recording this webinar so it can be accessed and viewed later on. So let's just give a couple minutes. All right, folks are joining. See if we can get to 20 attendees and then I will start it. 19. Well, we're leveling off at 19. All right, it looks like we're going to hold at 19. All right, well, thanks everyone for joining us, and happy Yuletide and all that jazz. Welcome back to the Interagency Working Group Ocean Acidification webinar series that is examining the science around acidifications, particularly within estuarine systems. And this webinar series is trying to be a precursor to a workshop that we hope to hold that will try to explore where there might be some real synergies between the federal agencies that can allow us to better understand how we can better serve the users and the people that need data and information products related to acidification within estuaries that fall within our various agency mission spaces. So the last two webinar series, we sort of set the stage for the state of the science on estuarine acidification. Today, we are going to be joined by two prominent researchers. Dr. Martha Sotula is the head of SCORP's Biogeochemistry Department, where she oversees projects related to the effects of climate change and anthropogenic pollution on acidification, hypoxia, harmful algal blooms, and eutrophication. Her research combines the use of observations, experiments, and numerical modeling to understand drivers and ecological impacts of these phenomena on streams, lakes, estuaries, and coastal waters. And she's worked extensively to link her science to management, serving as the lead scientist of the California State Water Resource Control Board, providing technical support to develop eutrophication water quality objectives for the state of California. She'll be speaking shortly after Emily Revest. So Dr. Emily Revest is a global change biologist at the Virginia Institute of Marine Science. And she works at the intersection between physiological stress tolerance and environmental exposure. Her research examines the physiological consequences of global and local stressors on marine invertebrates and coastal ecosystems. She leads a research program centered around the mechanisms underlying acclimatization and local adaptation to environmental change, with focus on anthropogenic stressors such as ocean acidification, ocean warming, and hypoxia, as well as natural environmental variability. She's examined several different systems. Of particular interest to us today in this talk is her work in the Chesapeake Bay, where she's looked at oysters and other bivalve effects. So with that, I'm going to go ahead and hand it off to Dr. Revest and let her present.
Thanks, Dwight, and thanks for the invitation to be here today. Let's get set up here. All right. So today I'm going to be talking about some recent work that we're wrapping up that was funded through NOAA's Ocean Acidification Program and their Regional Vulnerability Assessment category of awards. And we have been working on regional vulnerability assessment with respect to Chesapeake Bay and coastal and ocean acidification, focusing on aquaculture and restoration. And so I wanted to acknowledge the diverse group of collaborators and their expertise on this project. It's really been a phenomenal team, and we've made some really amazing advancements, like you'll see. This work also leveraged and built off of a previous OAP award focused on OA thresholds. And so hopefully you can see how work that's more focused on physiology can scaffold to inform these larger, more integrated projects. So in case you haven't been thinking about coastal acidification for a while, our study system here is Chesapeake Bay, where, like with open ocean and other systems, anthropogenic CO2 is changing chemistry that is affecting the habitat that organisms like oysters live in. We also have regional and local drivers like freshwater input that are exacerbating acidification through changes in pH and buffering capacity. And so it is a very complex and exciting study system for looking at impacts of acidification and how that translates to these roles of coastal organisms that directly interact with humans. So here in this project, we were interested in examining what ocean and coastal acidification might mean for oysters, their services, and human communities. And again, the Chesapeake Bay is an ideal study system for this kind of work because the coastal seascape is an ever-changing mixture of these drivers and co-stressors. In this map on the right that was produced through our project, this was our study region, which is the western shore of Chesapeake Bay in Virginia, and some of the bay tributaries. And here, plotted in color on the map, is omega calcite, with red indicating lower omega calcite. And the little black outlines that you can see are different aquaculture leases and restoration sites in our area. And so you can see that these natural gradients of exposure are laying onto this landscape of human use of these animals. And so whether we think about locations where the water is naturally more acidic in present day, or locations where water chemistry might be changing most quickly, this restoration aquaculture applications will certainly be threatened in some way. But we know that the animals aren't the only ones that are vulnerable. We need to think about humans too, especially when thinking about motivation of this work in terms of informing adaptation strategies and implementation. And we know that in general, people and communities differ in their sensitivity to change, and that includes people in communities that rely on oyster harvest and restoration. And that we also need to be thinking about planners and resource managers and users and what decision support tools they need to understand resilience in the system, as well as different social, ecological and social thresholds. And so as part of this project, and I'll get into this more in a moment, we built this website that users can use to explore the data and future predictions. So our first question, research question in this project is, how will acidification impacts to oysters drive sudden change in aquaculture and restoration? And to do this, a significant advancement to our field that came out of this project was the development of a finer resolution biogeochemical model that produced output at a grid cell size of just 120 meters. And this allowed us to resolve spatial variation in conditions within the footprint of an oyster lease, which is the first time we were able to do that. So if we want to be able to provide output that's useful for individual users as well as decision makers, we need to be producing it at a resolution that's relevant to their decision making. And so you can see here, this is the red outline of a single oyster lease, and you can see the individual blue squares that represent the grid cells. This model study region was focused on that western shore of Chesapeake Bay with the Rappahannock and York tributaries, where there are lots of oyster leases and restoration areas, and to translate how the different biogeochemical outputs in these grid cells would translate to changes in present day and future oyster growth throughout the region. We embedded bioenergetics oyster model that we had tuned for acidification impacts from our previous OAP award. So one of the products that I wanted to highlight here, and you can use the QR code to check it out, is a climatology that we produced that distills 4 decades of model results. This is at the 600 meter resolution, but something that was a product of this project and useful for a variety of applications. On this website associated with the DOI, you can access the climatology in the form of maps of surface and bottom conditions for each month of the year. So we have these ready-made visualizations, and we have the original gridded information as quantitative data that you can download. And in this atlas includes 27 environmental variables, including omega, the carbonate chemistry parameters, things like calcium, currents, and winds. So I encourage you to check that out and share it if you haven't seen it already. What we were using this refined model to do, Catherine Chaika, who was a master's student at BIMS, took this coupled model and she ran a series of scenarios to look at how acidification, the presence of co-stressors, and oyster responses look in the present day and in the future. And to condense her master's work to one slide, I'm showing you output from her present day run as compared to a mid-century combined future run that incorporates elevated atmospheric CO2, elevated atmospheric temperature, and reduced nutrient inputs reflective of meeting the EPA's total maximum daily load targets. And this was done using our 120 meter resolution model. So in this panel on the left here is showing the change in bottom omega calcite in each grid cell, comparing mid-century to present day. So acidification into the future is shown in the form of a negative delta, or these blue colors. And although there are a few places where we're seeing an increase, we're seeing a majority of cells showing a decrease in omega calcite going into the future. And we can use this to compare conditions in individual leases or restoration areas, identify places like this main stem shoal where the deltas are the largest. And she was curious how this would translate into effects on oysters. And so here in the panel on the right, I'm showing output as the difference in shell weight between present day and mid-century for a given grid cell for an oyster at the end of a 1-year deployment. And so we can see that in places, the results are shown in places where oysters were growing in present day. And you can see the blue color throughout, indicating that oysters in general will be growing more slowly in the future, and that the places with the largest negative impacts on growth are occurring in these the shoals of the tributaries. And these are places where there's really high oyster growth in present day. So sort of consistently, when thinking about both omega and oyster growth, the largest decreases happening into the future are occurring in places where those characteristics are highest in present day, which is interesting. But other thing I wanted to point out about this study that I can't highlight in the time I have here is that we did detect high spatial variability in growth differences. So high spatial variability in the impact of OA on growth between oyster leases or restoration areas. And this means that place-based predictions, like what this coupled model could produce, are really needed to inform future decision-making and adaptation planning. Another thing we wanted to get at with this part of the project is that, as I'm sure has been covered in this series and is well known, characterizing acidification in estuarine environments is very challenging, and there's been a lot of water quality monitoring, and Martha may get into this too, that has more of a legacy than acidification monitoring. And so we wanted to see, what if we, beyond the amazing contributions that the model can provide, what could we learn from the existing data? Without adding any additional measurements or anything like that, what can we learn from the existing data? And we have water quality data sets, ongoing water quality data sets in this study region. And so we just wanted to see what we could learn from that in terms of how exposure to acidification conditions varies among aquaculture lease areas. And so here is one figure highlighting results from that work, where we calculated this exposure metric that incorporates severity of an omega excursion below the thermodynamic threshold of 1 and the duration of that exposure. And here, positive values, this positive exposure term indicates conditions that are harmful for acidification. And so as we're going up the York Estuary from closer to the mouth, where the salinity is higher, to the headwaters, where the salinity approaches 0, you can see that this integrated exposure metric increases. And so we could determine the place in the estuary where we might start to see these negative conditions emerge. So sites with lower salinities are characterized by more acidification exposure. The other thing we're able to tell from this data is if this exposure differs by season, and we see springtime pop out as a season with relatively more exposure at any given place along the estuary. And that's because in springtime, there's more freshwater input to the estuary. And so at any given station, the salinity is going to be relatively lower than during other times of the year. So salinity in this system likely underpins seasonal variation in exposure. More detailed analysis, time series analysis of this data set does highlight biological processes as an important driver of omega variability, and that's something we can look more into in the future. It's still kind of an emerging part of this work. So moving on to some more of the social science components, Catherine's work showed how oyster growth might be affected at a given location in the future due to the combined impacts of acidification and other stressors. We wanted to see how that might translate to actual aquaculture production of oysters. So our collaborators took the biogeochemical output of our model, and we were able to get aquaculture production data for leases in Virginia, and used a random forest approach to look at the relationship between those biophysical stressors and production. And we found across 88 leases that that relationship was moderate, that the strength of that relationship in terms of explanatory power differs among growing areas and among seasons, with spring and summer being seasons with the strongest explanatory power between biophysical stressors and production. One advancement in this area was the development of exposure metrics where these metrics were based on calcification rate and of shell and growth rate of tissue of the oysters, and the integrated effects of multiple abiotic parameters and chlorophyll A on those things. And these integrated metrics were the most influential predictors of production, not the individual biophysical stressors. So this panel A here is showing you the average values of those exposure metrics, places where tissue growth is lower, but shell growth is higher, vice versa, across a larger study region. And panel B is showing these total production between 2005 to 2020 aggregated to water areas. We need to aggregate the data to protect the identity of individual leaseholders. People could be outed if we're showing lease specific information on a map. So with that, sensitivity is definitely something we take very seriously in this work. And this is where we developed this interactive geovisualization tool. And here's a QR code if you'd like to check it out. It's hosted at Oregon State University. This is where we were able to include the biogeochemical data from the atlas that I shared earlier in an interactive format so that users can click on a location and see data that's relevant to their location. So it integrates production data, aggregated production data, and these biological and physiological metrics that I've been talking about. And so they're able to see for themselves and investigate historical relationships between those things. We also used it as a research engagement tool. So our collaborators at Oregon State used it to gather information during interviews that they conducted with aquaculture and restoration community members in our area to indicate a place on the map where they observed a change in production in aquaculture or restoration performance and what they thought the reason for that was, or if they didn't know. So we've been able to kind of use it in this bi-directional way, which has been great. And so to kind of tie these things together, we wanted to look at the capacity of existing regulatory frameworks to enable adaptation in aquaculture and restoration activities. And to do this, we developed a social ecological system framework that's shown here in the conceptual diagram and used it to identify 3 ways that acidification and co-stressors is undermining livelihood resilience in the Chesapeake Bay region. Firstly, these stressors are deteriorating livelihood assets. Of particular importance to our users was access to shell, oyster shell, for use in their operations, and are pushing people beyond a threshold where they must adapt or accept cumulative risk of losses. And these stressors are also shifting the reliance on oyster seed from natural strike, where they put out shell and natural recruitment happens, to purchasing seed from seed producers like hatcheries. And that leads to inequitable outcomes for different actors, where small-scale growers are disproportionately affected negatively. And then thirdly, we identify these parallel dynamics through which both acidification and human decisions are diminishing the potential for shell accumulation in the system. So if we try to integrate this to think about what we could say in terms of adaptation pathways for people and communities in the Chesapeake Bay region, we think of adaptation as this lever by which people can control vulnerability. And there's really a need for targeted adaptation strategies that address specific vulnerabilities of different regions and different scales of operation, particularly for small-scale growers or projects that lack the resources to buffer against environmental changes. The temporal and spatial variability in exposure, sensitivity of the oysters, sensitivity of the humans that we have found in this project really highlights the importance of localized and even seasonal management approaches. Through this project, we hypothesized that people would be adapting to acidification impacts, but instead we found they're actually adapting to the social dynamics of seed provisioning. and this is relevant to both aquaculture and restoration. And acidification is instead a threat multiplier of existing social dynamics. And so looking into the future, developing social infrastructure and networks may make more of a difference in helping these communities adapt, more so than the development of private infrastructure like OA mitigation technology or the flexibility to change the location of operations. That isn't to say these things aren't important, but the social infrastructure side may be relatively more important. We did identify, though, that relationships with research between these community users and academic researchers does remain important for helping them identify problem sources and implementing associated remedies. Since this talk is being used to inform a workshop, I thought I would give a more informal conclusion, if I have time, to talk about some lessons learned through this work. One thing was that, working with this interdisciplinary team, it was the first time that as a natural scientist, I've partnered so closely with a social science team. And this kind of collaborative project, I think, is really what's needed to address these issues of coastal resilience in the context of coastal and ocean acidification. But of course, the more diverse your team is, the more challenging communication and staying on the same page is. So being in frequent communication was really important, and really integrating those end user communities, whether it's aquaculture, restoration, or other things, throughout the entire lifespan of the project, from conceptualization to product implementation, is really critical to make sure that the research we're doing is useful and usable to those communities. We worked with Karen Hudson, who is an aquaculture extension specialist at BIMS. So that's who I'm referring to here with Karen. She is one of those linkers that gave us access and served as a bridge between what we were doing and the end users that we're trying to serve. Longer term project timelines was really helpful for this, because for a lot of the end users that we work with, the issues that they're dealing with are more immediate and stochastic, and they come and go. The pandemic affected our project and access to people, and also the priorities of people that we were working with. So having a longer term support timeline to really build those relationships can lead to greater impact and greater diversity of impacts in the future. There's of course more to explore with this work, and we're really excited that to have the potential opportunity to examine comparisons within our region. We focused on one area in the region for this project, and we're able to make large advancements because of our focus. And now we would have the opportunity to compare communities with different social vulnerabilities, different bivalve aquaculture species, different emission scenarios, and to hopefully with another pandemic not in the immediate future, to do more co-production of those visualization tools and model outputs with end users. So I think I'll leave it there. I really look forward to your questions and discussion at the end. I'm not sure if we're taking some questions now or waiting until the very end. Thank you.
Thank you, Emily. Yeah, that's fantastic. I think we're going to hold questions to the end, but by all means, if everyone could please enter your questions you might have into the chat. I have a few of my own I might be asking too. Really good stuff, Emily. Thank you so much. And with that, I think we're gonna drift over to the West Coast a little bit, and take a look at some of the work Martha's been engaged in. So take it away, Martha.
Okay, so I'm assuming that I've got the right screen up. Is that correct, Dwight?
Yes, yes, you do. Yep, looks
okay,
great.
Excellent.
Yep.
Okay, well, thanks so much for the invitation. Welcome everyone, and I'm really proud to present the work that my colleagues and I have been doing for the last 10 years. on the West Coast, thinking about how we can increase resilience to acidification and hypoxia in Southern California coastal habitats. And specifically, you know, in thinking about your workshop today, what are the ingredients that I think are important for how we can start to accelerate some of those management decisions? And for context, for those who know me, maybe on a more personal level, I'm actually an estuarine ecologist and biogeochemist, and I've done much of my own science, you know, kind of primary science in estuaries. But when I was asked to give this talk, you know, and I thought about what data do I have on acidification in Southern California estuaries, and just for context, there are about 40, you know, small to medium size along our Southern California coastline. The answer was almost zero. You know, very, very little acidification data, lots on hypoxia. So for the story today, what I'm going to do instead, I'm going to talk about some of the work that we've been doing in our nearshore ocean environment, because I think it provides a really nice, I guess, story arc and things for us to think about. What do we need to really implement for estuaries? And this is part of a long-term multi-PI science, more than 20 principal investigators, 5 institutions. And so I'm really here as the, you know, I guess, science communicator in chief. Most of the work is done by other people. So when I sort of listened to all of the, you know, the previous webinars leading up to this one, and you can also sort of hear this in Emily's talk as well, I think what we're really facing is multiple challenges in trying to accelerate management decisions to alleviate acidification stress on coastal habitats. We've talked about multiple stressors, the tremendous amount of geographic variability, whether it's within an estuary or among estuaries or among our coastlines. So absolutely a one-size-fits-all approach will not work. But yet, at the same time, I think we have this urgency to act quickly, and that's, you know, because we have done those model scenarios to show that, you know, that by the turn of, you know, 2100, we're looking at a catastrophic loss of our habitat. And so that urge to act quickly, though, is confounded by lack of biologically relevant water quality goals. So we don't really have clarity in what we're trying to manage to. There's a scarcity of monitoring and research that would support those actions across our estuaries, multiple jurisdictions, and often we can get in the way of one another. And then finally, especially because some of the solutions that we're proposing are sort of starting to pinge on control of pollution inputs, that there's a lack of buy-in or support from those regulated communities. And so that all presents, I think, a formidable barrier. And so what I want to talk about today are, you know, kind of, you know, maybe writ large, a suite of scientific ingredients to what a solution could look like. And what I'm going to do is, as I proceed to these slides, I'm going to just give sort of the anecdotal description of these solutions in this case study of what we've done, looking at the potential control of anthropogenic nutrients and improving resilience to acidification and hypoxia in the Southern California Bight. So I think where we all understand this, and, you know, and it's beautifully demonstrated in Emily's talk, and the fact that you guys have an interagency working group, is that, you know, I think the first place where we need to invest resources is really in the science and management conversations in a joint and integrated way that allow us to explore solutions with broad partnerships. And in California, the way this has played out is that we had a West Coast OH science panel that was pulled together by the 3 states, Washington, Oregon, and California, that set out a set of management recommendations that the California Ocean Protection Council in particular really took to heart and began investing in strategies to mitigate the effects of climate change. And reducing anthropogenic nutrient inputs was identified as one strategy, but by no means was it a foregone conclusion that such a strategy could be useful, especially because the reigning paradigm on the West Coast is that we have such a strong upwelling along our coastline that, you know, where could reducing anthropogenic nutrients ever matter? And so that was the mindset, especially when I began my career out here about 20 years ago. And so since the time period that the OPC began to, essentially develop its strategic plan, they have partnered with NOAA and made strategic and sustained investments in the science to have place-based conversations about solutions. So again, no one size fits all. And they're doing this through these regional cooperative partnerships of stakeholders that are willing to share their own data and that have already been engaged in using science to support policy decisions. And so this is a big part of how we do business, and we're not alone. Again, Emily provided a beautiful example of this in the Chesapeake Bay. So the second ingredient then is that there needs to be sustained investments in both observations and numerical models. And when we talk about making decisions about what specifically we want to do to improve resilience, models become a key component of how we support and help the community understand and get behind certain actions that represent the consensus for how to move forward. And so for us in Southern California, and actually on the Pacific West Coast, NOAA provided, along with the Ocean Protection Council, some key funding to develop the regional ocean modeling system, coupled to the biogeochemical elemental cycling model, which is an intermediate complexity nutrient and biogeochemical model that allows us to basically predict how algal blooms that are sourced from natural as well as anthropogenic or terrestrial sources of nutrients can proceed through biogeochemical cycles that result in the consumption of oxygen and pH. And that when we couple or we force that model with inputs from rivers, from ambient CO2, the ocean outfall discharges that are the primary way in which wastewater is released in our Southern California environment, as well as atmospheric deposition, then we have a way for us to use this model as a place-based tool to look along the coastline and say, okay, where can we start to understand that those anthropogenic nutrients may make a difference? So this model was a long time in the making, 2 primary publications shown here. But what I want to emphasize is that, it's really built on the backbone of about 30 years of research in the physical modeling, coupled to a wealth of primary data through regional monitoring partnerships, CalCoffee and the Southern California Bite program that our institute manages, that really represents the backbone of the types of data that you would need to credibly develop and validate such a model. And so the Southern California Bite ended up being the first case study of nutrient effects because of the wealth of the data. And not coincidentally, there's also a coastal population of about 23 million people with 19 ocean outfalls and about 75 rivers rather, that really represents a place that, hey, if we're going to see an anthropogenic signal, we ought to see it here. And so these data then became a really important means for us, not only to set up the model, but also really to drive a lot of validation studies of that same model. Here's just an example of some data model comparisons done by regions along the coastline using municipal agency data. And it's also really key data for biological threshold development and validation. So how is ROMSBEC used to make predictions of the biogeochemical effects of terrestrial nutrients? Well, in the most basic way that we do this, we do a change assessment of 2 simulations in which the top is a full sort of realization of all the anthropogenic nutrients coming into the coastline. And that is subtracted, from that we subtract an ocean only, or essentially what is our best representation of a natural scenario without any influence of anthropogenic nutrients. And so we use this type of approach to basically identify what is the effect or attribute the effect of anthropogenic nutrient inputs, and then really start to understand or investigate the drivers of how this results in an additionality of acidification and hypoxia. And what we've published over several sets of years at this point is that the models predicting that anthropogenic nutrients lead to a persistent increase in algal biomass, particularly along the coastline, and that it's a chronic year-round effect in which we're essentially demonstrating or showing how that's trapped in the upper mixed layer. And I'm showing you this cross-section throughout the central area of the bite. And so what you see immediately in this change assessment diagram is we start to get, we have this persistent production where a lot of the rivers and the outfall pipes are coming into the coastline, but then that appears to be some sort of trapping of that organic matter in the region of the central bite. And so then, following through the story, what we're finding then is that the model's predicting a seasonal offshore subsurface acidification and oxygen loss. And the mechanism for that loss is actually driven by recurrent eddies. And so the idea behind this is that you have this cross-shore currents that develop a set of mesoscale and submesoscale eddies that essentially are trapping both inorganic and organic matter within these ephemeral or current eddies, and that in those locations then we have an enhanced mineralization, remineralization that's leading to subsurface oxygen and acidification. So, hold on, let me back up a second. So, okay, so we can predict the number of moles of change of oxygen or the changes in DIC, but ultimately what managers really wanted to know were what were the effects on biology. And so we saw this coming, and part of our science plan was a concerted effort that really start to try to understand what are the thresholds of biological impacts. And for reference, yes, the California Ocean Plan has existing pH and oxygen water quality criteria, but particularly if you're interested in acidification, those pH water quality criteria are not biologically relevant. And so the approach that we took and thinking about how we would go about making predictions for the biological effects is to quantify the amount of habitat compression that would result from changes in pH and oxygen regimes. In terms of, since this is the coastal ocean, we're actually looking at the change in the habitat thickness or volume over time for different species. And so in order to tackle this, and we understood that acidification is the new science, and we want to make headway there, but we're also going to co-develop at the same time a similar sort of parallel set of activities for oxygen and try to move them both forward. And so we worked on a set of tools through different funding sources to come up with essentially thresholds or indices to predict biological effects of acidification and hypoxia on California current species. For acidification, what we did is essentially, again, new science. So we focused on an expert synthesis of existing research, looking at over 135 published laboratory and field studies to be able to generate through a set of expert workshops, a set of thresholds for pteropods, echinoderms, and decapods, ranging from sublethal to lethal effects. On the bottom graphic here, that just shows you sort of an example of the type of syntheses that we did as starting points for us to consider what we may be able to use. On the oxygen side, we took a different tact because what we understood is that what really matters to the species for oxygen is essentially what their is not only the supply of oxygen, what's in the ambient water, but the species-specific demand for oxygen that is that's also a function of temperature and depth and other things. And so what one of our collaborators have been leading on, Curtis Deutsch, is the development of something called the metabolic index that essentially allows you to make a prediction of what would be the level of oxygen that would be protective versus lethal to species, depending on the amount of supply plus the temperature regime of the water column that you're finding it in. And so we did this both as a specific California current case study for northern anchovies, as well as develop additional metabolic indices for over 15 benthic and pelagic and commercially important species in our region. So to kind of distill this down, then what we did is that we took these two approaches and then we tried to essentially bookend them in terms of the potential changes that we may find as we apply this back to the model. For what would essentially be levels that would be protective of healthy populations versus any potential lethal or reproductive effects for both pteropods and for acidification and oxygen for northern anchovy. And so this is the work of Christina Frieder that she just published this fall. And that where our model is showing essentially in the same area that I showed that bullseye of kind of respiration and oxygen and pH loss, is that the model is predicting a habitat compression of sublethal thresholds seasonally for about 3 or 4 months annually, and that the region of that recurrent habitat compression is occurring mostly southeast of the Catalina Islands offshore and is thousands of kilometers in size. So now we've taken that chemical output and translated it to essentially what is the potential for biological impacts, and showing those results and look actually very similar for the calcifier change in habitat capacity, as well as the change in aerobic habitat capacity for northern anchovy based on these two sentinel species. Okay, so managers then began to take this work seriously. And as they began to have conversations about what they may want to do about it, the stakeholders also really got quite a bit more engaged, in particular the regulated community, and questioning whether or not the model should be believed, or is the model, are these model results credible, and are they management actionable? And so we really then kind of pivoted and over the last couple years and have been working very hard on sort of two different pieces and thinking about how do we move this forward, is that if we understand that a key component to really moving towards some management decisions about how to increase the resilience of our ecosystem, that if we're going to fight over the science, it's going to really cause us to lose years in the bargain. Perhaps, you can have lots of conversations over the policy decisions, but we're hoping really to get a lot of scientific consensus around our findings so that we're not fighting over the science. And so kind of to really, put a fine point on that, we're really doing two things. Number one is that we're working on quantifying and communicating uncertainty on our model predictions. Both in terms of sort of an ongoing and transparent model skill assessment. Here's an example of a tailored diagram we have from our multiple parameters that the model is predicting. And then we're also really trying to, force ourselves to identify whether or not the quantity, if the magnitude of the signal that we're predicting, which is essentially the change in the model that's due to anthropogenic nutrient inputs, is greater than the data model predictions. And so I'm showing you an example of, here's our control scenario, our natural scenario, and then our anthropogenic scenario, and then the difference between essentially the observations and the model in the region of this particular sort of donut hole of of high respiration. And so what we're trying to essentially assess is whether or not the change or the difference between data and models are essentially smaller than what we're predicting to be the effect of anthropogenic nutrients. And so we're starting to hone in on these methodologies to really start to communicate this and to provide some sense, policymakers and the regulated community with some sense of to what degree they should think that these results are credible. The other thing that we've been doing is really trying to support a stakeholder-funded expert panel review of our numerical modeling tools. And so they took it upon ourselves with full cooperation of the regulators and our science team to bring on 6 international experts that responded to a set of charge questions from the steering committee to basically opine whether or not this model should be something that should be taken seriously in terms of its scientific findings. And they just came out with the results. I threw out a few anecdotes here, which basically provided a green light for the model to be taken seriously. But they also provided over 40 recommendations for how we should be working with them to improve stakeholder confidence in the scientific toolkit. And the community is responding by thinking about how do they prioritize those recommendations and how do they work together to get them funded. So on to this sort of 6th ingredient. So we are starting to think now, and we've already been starting to use the model for different types of applications. And so as we pivot and start to think about that, I think we need to have an open mindset about what sort of solutions we should be looking at and having flexibility and potentially what that solution could be. And so, yes, we've already been doing lots of scenarios that look at nutrient management in the bite. But the reality is that as we start to think about wastewater upgrades, they're likely to go in hand in hand with reductions of the volume of wastewater that's going out to the bite due to the intense interest in potable water recycling. So they're actually going to be recycling water from wastewater and eventually turning it into something that people can drink in order to increase the region's resilience to another effect of climate change, which is drought. And so one of the questions that we answered or began to try to answer with our model is trying to understand, as we begin to reduce the volume that results in more highly concentrated wastewater discharge, how does that affect the outcomes that we're seeing relative to some amount of nitrogen reduction that could be occurring through these plant upgrades. And so I'm just, I'm providing the reference. If you're interested, I won't spend any more time talking about the results of that. But you can see the model now begins to pivot and become a type of tool that you can use for decision support in a sort of what-if scenario without actually spending a dime on the actual solution itself. Another example of this is we've also really, through additional funding from RPE, have been developing aquaculture farm modeling tools that would actually allow us to simulate what is the effect of growing kelp in the Southern California Bight, a region that has not historically had a lot of aquaculture, and how would that change relative to kelp alone or doing this as a co-culture with shellfish. And so this is another potential solution. And the question is it important that we do nutrient management alone at the tune of billions of dollars, or can there be some artful combination that considers essentially where this would make sense along the coastline, in which you could be looking at some combination of aquaculture that could be helping to polish or additionally provide other important system benefits. So kind of to wrap this up, we are, really pursuing a lot of different types of science and directions that I think would make a fun conversation for us, at the workshop and beyond. Of course, a strong focus on those panel recommendations to increase community acceptance, namely improved observations, more skill assessment, data visualization, and more. We're actively engaged in discussing what are the next set of scenarios the management community would like us to run to support decision-making on nutrients and potentially other solutions. We're engaged in moving the numerical model inshore into estuaries to downscale and incorporate some of the new processes that are important in those types of systems. We're also really thinking very hard about how to move forward with multiple stressor biological indices, because ultimately we know that univariate thresholds, especially for places like the ocean and in environments that have significant warming, will not be sufficient. And in order to do that, we're coupling experiments as well as increased monitoring of both biology and chemistry using metabar coding and OA-specific biological indicators. We're expanding this work to new geographies. We already have some new work that should be coming out within the next year in the San Francisco and Monterey coast. And then finally, we're expanding model applications, looking at how we start to model the effects of marine carbon dioxide removal as a solution, looking at toxic HABs as another endpoint of concern, and then finally really starting to do some of the very fine scale riverine and outfall plume studies that our stakeholders think are important to do in order for us to really gain their acceptance for the types of decisions we think we need to support, particularly as we move inshore. So with that, I will leave you with sort of the laundry list of ingredients and acknowledge our funding sources, and especially all of the folks that work very hard to put all this work together. And I'll stop there. Thank you very much.
Thank you so much, Martha. Fantastic talk, very insightful. I want to make sure we have a few minutes to maybe address some of the questions in the audience. I think we'll start. I know that Emily has a hard stop at 5 p.m. So we'll ask your questions first. We did have a question that came in from Beth Turner, and she's asking, do you have an idea of who might be accessing the Atlas and the geovisualization tool, and how are they being used outside of the project itself? Oh, you're muted, Emily. Sorry.
Thank you. Those are great questions, and both for release this calendar year. So I think it remains to be seen the ways in which they're used in the future. One of the downsides of doing a project during the pandemic is we didn't get as much end user engagement as we had originally intended in that project. So we're hoping to build on that through a future project to really do a lot more of that workshopping with a visualization tool and more sort of hand delivery of that in different settings. I think there's also a need for consolidating OA resources, particularly at a region level, so that there aren't these individual project-based tools that are harder to find, and hopefully more of a repository, like a go-to for like a dashboard for the aquaculture community. So that's something we're thinking about as well. For example, Margie Friedrich's group produces nowcasts and very short-term forecasts for a variety of parameters in Chesapeake Bay, including acidification parameters. And so, co-locating that with social threshold work that we would do, or these other kinds of visualization tools, and a way to interpret that. So a lot of feedback that we've gotten from the industry is, okay, it's, people deliver maps, or you have real-time observations that are available to anyone in the public online, but that doesn't come hand in hand with an explainer that says, and this is what it means for oysters. If the pH is this or the saturation state is this, you know, is that good for oysters or bad for oysters, or whatever other, you know, kind of interpretation you want. And so, just like, you know, Martha mentioned, we need coupled biological and chemical monitoring. We also need coupled share out, and so those are ways we want to enhance our products in the future.
Thanks, Emily. And might that be in the form of maybe some committed product training workshops that would happen, you know, directed to the industry folks? And if so, where would that happen? I mean, what would be a venue for that?
Yeah, we have a lot of opportunities for engagement in our area. So we have our Virginia Aquaculture Conference, for example, that often has those kind of workshops, or you can easily meet with people one-on-one or in a group setting and do that kind of work. And BIMS has other advisory committees related to shellfish aquaculture. So we have that infrastructure, but we need the project support to do that co-production, and so that's what we're trying to continue with.
Excellent. Thank you, Emily. I don't see any other questions for you in the chat. So I know you have a hard stop, so you're welcome to go.
Yes,
For those
you.
who want to, who can stay on, if you want, maybe a few questions for Martha. I've got one quick one for you, Martha. Or maybe it's not quick, but so you were beginning to get into it in the presentation itself, and it's making me think back to the conversation we had previously before this talk about you've got these nuanced integrated products, you know, like habitat compression that ultimately needs to lead to some nutrient loading target for the state. How receptive has the state or the federal entities been to maybe revising their guidance or their guidelines, you know, based on these products? And I know you spoke to it. Part of it is trying to sell the quality of the product, but I mean, if we can be confident in that the model is more accurate, do we think we have a pathway to begin to influence and modify those state and federal agency guidelines?
That's a really good question. So, and I think there's a lot of different pieces of that. So the first thing is that, you know, I think that the scientists should characterize what we've done is sort of early in the game. And what I mean by that is right now, most of the published science, with the exception of maybe one or two pieces of work, have really been kind of bookend scenarios in which, you know, they're sort of extreme, in which the anthropogenic nutrients are all in or are all out. And, you know, we're just starting to do some of the work that really helps to understand what sources and what pathways could be looked at in terms of at least understanding their relative contribution to that overall problem. So, you know, I think that there's more science to do. That's the first thing. The second thing is that you know, we really need to make a connection between essentially what are the existing water quality criteria for the ocean, all the way to what would, you know, if the state agencies or the federal agencies want to, you know, essentially make a management decision, how would that link back to some sort of nutrient reduction? And so there would be, you know, conceivably a lot of work to be done, because right now no one's really talking about using those existing criteria. The state of California, based on the science that we've done, is actually on the books for having a project to develop new ocean acidification water quality criteria. And that's something that they are interested or intending to do. And so there would need to be more work to really make that linkage between whatever goals they intend to use all the way back to the the nutrient reductions that would be required to meet them in order to satisfy those criteria. And so there's quite a bit that's still up in the air. And I think that what we're trying to do, I think first, at least among the scientific community and you know, in SCORP in this leadership role, is to really make sure that people are getting comfortable with those decision support tools so that we're not arguing about the science, and that the stakeholders or the regulated community can really have a productive and straightforward conversation with the regulators about, you know, what they intend to do and what's next. And I think that, you know, these are all public agencies. They're great environmental stewards, and so they're just trying to do their due diligence in making sure that, you know, that the decisions that are being proposed are really, you know, based on solid science.
Well, my hope is they don't reinvent wheels, and that you're very much in the room in those conversations, and they don't kind of run parallel to all the effort that's been done to date to try to reinvent how to do this. So I appreciate the great work that you're doing.
Thanks. Thank you very much.
And let's see, are there any other questions? I don't see any more coming in. Let's see. Oh, let's see. Stuff is starting to come in now. I think they're just thank yous. So I'm a little unclear. This works. Okay.
I can't see the chat.
No, sorry. I can see it. It looks like it's good. Jesse Turner just says, great presentation. Thanks, everyone. So that's great. All right. And with that, I will bid you all adieu and a happy holiday season to you. Thank you so much, Martha. And the IWG will.
Thanks for your time, you all. I appreciate the opportunity.
Yeah, it's fabulous. So we hopefully will take all this as great fodder into the workshop we have planned, hopefully coming up in the fall of next year. So thank you so much. Take care, everyone.
Have a great day. Bye now.